This protocol is significant in that it allows for high-throughput ChIP-Seq analysis of primary samples to better understand cis-regulatory mechanisms and its implication on disease. The main advantages of this method are the reproducibility of the data and the drastic reduction of hands-on lab time.
For chromatin shearing, add 70 microliters of fresh, room-temperature complete lysis buffer to a glycine-fixed, snap-frozen, and thawed cell pellet of interest for a 1-minute incubation at room temperature, followed by 1 minute of resuspension without bubbles. Incubate the sample for another minute at room temperature before placing the cells on ice. Transfer the resuspended pellet into a 0.65 milliliter low-binding tube on ice, before loading the tube onto the tube holder of a sonicator. Fill any gaps with balance tubes containing 70 microliters of water, and let the cells stabilize in the water bath for about 1 minute before starting the sonication. After 3 cycles, remove the samples from the sonicator for gentle vortexing, and pulse spin the tubes before placing them back into the holder. For histone modification, add 100 microliters of complete TC1 buffer to each tube of 2 ChIP 8-tube strips, and transfer 20 microliters of each of 16 chromatin sheared samples into individual tubes of the ChIP 8-tube strips. Wash the chromatin tubes with 80 microliters complete TC1 buffer, and pool the washes in the appropriate tubes of the ChIP 8-tube strips